Okay, hello to all of you. Let's continue discussing chapter number 7. And let me go into slideshow mode. Okay, so till now we have discussed passive transport. And now we are going to talk about active transport. So basically, as we discussed in the passive transport, that it's a movement of molecules from high concentration to lower concentration. The molecules or the solutes, they move down the concentration gradient. So which is a spontaneous process, which is a natural process. It's a natural tendency of molecules or solutes to move from high concentration to lower concentration. So this process does not involve any energy. So that is why it's called passive transport. The next kind of transport mechanism we are going to discuss is called active transport. So active transport is a transport mechanism in which the molecules or the solutes, they move from low concentration to higher concentration. So that is why in this process there is a requirement of energy, because here the molecules and the substrates, they move against the concentration gradient. So this is an energy requiring process, and this process needs energy, and this energy comes from ATP hydrolysis. And in this active transport, the typical example of active transport is a sodium potassium pump in animal cells, and there's a proton pump in the plant cells. So we are going to talk in detail about sodium potassium pump, which is a typical example of active transport in animal cells. So let me explain you how this pump works. Okay, so basically what do we mean by a pump? Pump means a protein. So remember that in active transport, there is always involvement of transport proteins, and the proteins which are involved in active transport, they are called carrier proteins. So basically this is a protein, and this protein has 3 binding sites for sodium ions. Okay, so this is a protein. You can see these are the 3 binding sites for the sodium ions. So in the sodium potassium pump, which is the active transport, the molecules move from low concentration to higher concentration. So inside the cells, there is less concentration of sodium ions, and outside the cell, there is high concentration of sodium ions. So through this protein, the sodium ions are moved from inside to outside. Inside the cells, there is low concentration of sodium ions, and outside the cells, there is a high concentration of sodium ions. So the sodium ions are moved from low to high. So this is active transport. And also through this protein, potassium ions, which are low outside the cell, they move from outside to inside. So inside there is a high concentration of potassium ions. So the potassium ions, they move from outside to inside. Outside, the concentration of potassium ions is low, and inside the concentration of potassium ions is very high. So again, potassium ions, they move from low to high concentration. So sodium and potassium ions, they are transported through this protein against the concentration gradient, and this process needs energy. That is why it's called active transport. And we will see that the energy for the transport comes from the hydrolysis of ATP. So look at this protein. It has 3 binding sites for sodium ions. So sodium ions, they bind to this protein, and when that happens, followed by the binding of sodium ions to these 3 binding sites, there is hydrolysis of ATP. ATP hydrolyzes, and it breaks into ADP, which is adenosine diphosphate, and phosphate. And this phosphate group, it attaches to this protein, and this protein becomes phosphorylated. So the protein gets phosphorylated, and when the protein gets phosphorylated, its shape changes. So once the protein gets phosphorylated, its shape changes. So once its shape changes, you can see how the shape of this protein is changing. So when the shape of this protein changes, in that shape, this protein cannot keep sodium ions with itself. It has to deliver these sodium ions outside the cell. Okay, and in this shape, this protein can also bind to potassium ions. So there are 2 binding sites. There are 2 binding sites for potassium ions. So in this shape, it is going to deliver, this protein is going to deliver sodium ions outside the cell, as well as it can also bind potassium ions to itself. And followed by the binding of potassium ions to this protein, followed by that there is a process called dephosphorylation. This protein becomes dephosphorylated. This protein loses the phosphate group. And when that happens, the shape of protein changes. So when the protein becomes dephosphorylated, the shape of the protein changes, and this protein gets back to its original conformation, and in this shape the protein can no longer bind potassium ions, and it delivers the potassium ions inside the cell. But in this shape it has again the binding site for 3 sodium ions. So this is the way through this protein there is pumping of sodium ions from inside to outside and of potassium ions from outside to inside. So this is a typical example of active transport in animal cells. So this is all what I just said. So this basically shows overall all the 3 processes we have discussed: active transport and passive transport, in which is diffusion, where molecules move from high concentration to lower concentration. No protein is involved, no transport protein is involved in this process. And second is called facilitated diffusion, in which there is a movement of molecules from high concentration to lower concentration through the transport proteins. So these are channel proteins, these are carrier proteins. So this is facilitated diffusion. This is active transport. In the active transport, there is the movement of molecules from low concentration to high concentration, which is against the concentration gradient. So this process needs carrier proteins as well as it needs energy, and that energy comes from the hydrolysis of ATP. Okay. Another thing I would like to tell you at this point is there is another active transport mechanism which is very common in the plant cells. It's called the proton pump, and it's also in fungus and bacteria, proton pump. So basically it transports protons against the concentration gradient. And there are some terms which we need to know because sodium potassium pump and the proton pump, it is basically transporting all the ions, and because of these ions there are some charges involved. So let's talk about some of the terms. First of all, what is membrane potential? Membrane potential is the voltage across a membrane. Voltage is created by the differences in the distribution of positive and negative ions across the membrane. So the cytoplasmic side of membrane is negative in charge relative to the extracellular side. So two combined forces, collectively called electrochemical gradient, drive the diffusion of ions across a membrane, and those two forces involve a chemical force, that's the ions concentration gradient, and second is the electrical force, which is the effect of membrane potential on the ions movement. So chemical force is because of the ions concentration, and electrical force is because of the charge of the ions. So these two forces together drive the diffusion of ions across a membrane, and this is called electrochemical gradient. And electrogenic pump is the transport protein that generates voltage across the membrane, because it's involved in the transport of these ions across the membrane. So we have already discussed what sodium potassium pump is and what is proton pump. So this is the proton pump. You can see the protons are in less concentration on this side, more on this side, and these protons are being transported through this protein from this side to this side against the concentration gradient, and it involves a carrier protein, and it involves the hydrolysis of ATP. Next transport mechanism which we need to know is called co-transport. So co-transport is a transport mechanism in which two solutes, they are transported together. So let's see how it actually works. So in co-transport, basically there are two steps. First is, you can see here is the proton pump. So here the protons, they are being pumped by active transport from low concentration to higher concentration. So this is your proton pump, this is a transport protein, and these protons, they are being transported from this side to this side, from low concentration to higher concentration. And as a result of this transport, what is happening is the protons are getting accumulated on this side. So there are more protons on this side, less protons on this side. Okay. So as a result of that, the gradient is formed. So because the protons are more on this side, now the protons are less on this side. So these protons can move from this side to this side along the concentration gradient, and this is a diffusion, and this is a facilitated diffusion because this protons will be pumped through this transport protein, but this process does not need energy. But when these protons are being transported or being diffused from this side to this side along the concentration gradient, they can help in the active transport of another solute. Remember that they can help in the active transport of another solute. Like for example, here is sucrose. Now sucrose is being moved from this side to this side. Here is a low concentration, here is a high concentration. So these protons, they're moving from high concentration to low concentration. So this is a natural flow. This is a spontaneous process. This does not need energy. But along with the transport of these protons, the other solutes can move through this protein, and their movement is against the concentration gradient. They can be moving from low concentration to higher, which means when these protons are being transported passively, they can help in the active transport of another solute. And this active transport can be in the same direction as these protons, or it can also be in the opposite direction. So this is called co-transport. So co-transport occurs when active transport of solute indirectly drives the transport of other substances. The diffusion of an actively transported solute down its concentration gradient is coupled with the transport of second substance against its concentration gradient. Okay, the last transport mechanism we're going to talk about is called bulk transport. So it's a transport mechanism by which the big things, big molecules, macromolecules like polysaccharide proteins, they cross the membrane. So this is a process which also needs energy. And usually, this is a process in which big molecules, they cross the membrane via vesicles. In this, basically vesicles are formed, and it needs energy. And there are two different kinds of transport mechanisms we are going to talk about. One is exocytosis and second is endocytosis. In the endocytosis, there are three different mechanisms we will talk about. They are phagocytosis, spinocytosis, and receptor-mediated endocytosis. So exocytosis is a process in which something very big, it's basically leaving the cell. It's moving from inside to outside the cell. So in exocytosis, transport vesicles migrate to the membrane. They fuse with it and release their contents outside the cell. Many excretory cells use exocytosis to export their products. And in the endocytosis, it's basically when cell is taking in something big inside. So it depends what the cell is taking. If the cell is taking something which is solid in nature, it's called phagocytosis. It's called cellular eating. If the cell is taking something like a liquid, it's called pinocytosis. It's like a cellular drinking. And third one is receptor-mediated endocytosis. So here you can see this is phagocytosis. Something big and solid is entering into the cell, and you can see how the cell is forming this kind, how the membrane is getting invaginated and it's forming a kind of a food vacuole. This is pinocytosis. Here are some fluid molecules. You can see how the membrane again is invaginating and it is forming this kind of a vesicle, and how it is entering inside the cell. And this is receptor-mediated endocytosis, in which these molecules, these solutes, first of all, these molecules or these solutes, they first of all bind to these receptors. And then this membrane invaginates and it forms this kind of vesicle. And these are usually coated vesicles. They are coated with some special kind of proteins. And you can see how this vesicle is being formed. So this is called receptor-mediated endocytosis. And here is one example of receptor-mediated endocytosis. So human cells, they use this mechanism called receptor-mediated endocytosis to take in cholesterol. So cholesterol, which is actually combined with some proteins, and these proteins are called lipoproteins. So our cells, the human cells, they use this mechanism to take in cholesterol inside the cells. But there are some people who have either missing or defective receptor proteins, who have either defective or missing these receptor proteins, and these are called LDL receptor proteins. So what happens when these people don't have these receptors for binding to the cholesterol, then what happens is these people have a high concentration of cholesterol in their blood, because the cells cannot take the cholesterol inside them. So this disease is called hypercholesteremia, and because of the defective or the missing receptors, cholesterol cannot be transported inside the cells, and as a result of that, there is a high concentration of cholesterol in the blood, and this results in a disease called hypercholesteremia. So that's all in this chapter. If you have any questions, please let me know.